Assalamualaikum warahmatullahi wabarakatuh. Today I would like to summarize about the various stages of tissue culture based on the Shomer's biology video. There are six stages of plant tissue culture, which are embryo culture, bud culture, pollen anther, callus culture, protoplast culture, and meristem culture. The first, meristem culture. Meristem culture uses meristematic tissue or apical meristem, which is located at the shoot of plant. For example, part of plant shoot is taken and deposited in solid nutrient media, which consists of sufficient nutrient which is needed by the plant tissue culture to grow. It will be let to grow until a bud formation and generation of plantlet. The plantlet then will be transferred into the soil for the generation. The second one is bud culture. It uses explants from other plants or meristematic tissue. Part of the tissue is sliced and deposited in the nutrient media. Sufficient period of time and nutrient will be given until the proper formation of shoot and root. Complete one will be hardening and ready for the planting in soil. The third is embryo culture. It uses seed tissues of embryo cotyledon. The slice of tissue of the cotyledon will be placed in the nutrient media for plant development. After a certain period, there will be a formation of rooting and shooting. The whole plantlet will be taken from the media and placed into the soil for the generation. The fourth is the pollen or anther culture. It uses pollen or anther of the flower plant. Filaments will be removed from the anther and placed either in solid media or liquid media culture. It is necessary to provide a large surface area for the culture, as this type of culture needs more space for the formation of plantlet. The plantlet will be generated and will be transferred into the soil. The fifth is callus culture. It uses meristematic tissue and placed in the nutrient media. The cell will be dividing rapidly and will form a cluster of cell aggregates. This cluster of cells is totipotent, which is the cell has the capability to express the entire genome required to produce an entire plant. The cell cluster will undergo some process which are from heart-shaped formation, elongate to become torpedo, and then will invade in the solid media. The process will be further with development of seed leaf and roots, which require a specific hormone. Callus culture requires appropriate solid medium with both an auxin and cytokinin in correct condition. As we know, cytokinin promote the growth of shoot, and the auxin promote the proliferation of roots. 2,4-D, the common hormone commonly used. Type of hormone and concentration used are different from the type of species. It is useful for the polyploidy plant as they cannot able to generate themselves. And 6 is protoplast culture. In this culture, we use small part of the leaf. It will first undergo sterilized treatment. Before we transfer the the tissue culture, we have to prepare a protoplasmic solution where we will be break down the leaf in the small part and will transfer into the test tube and centrifuge until it will produce a solution which is called protoplasmic solution, or in the other word we call it as the extract of the protoplasm cell. Cellulose and pectinase are used as an enzyme to break the cell wall of the uh, plant tissue. That's all from me. Thank you.